Good morning, everyone. I am Rima, and I've been working as a postdoctoral researcher in the lab of Professor David James for about a year now. I apply bioinformatics and systems biology tools in the research of diabetes and obesity here at the Garden. And as you can see from this timeline, diabetes is a complex and progressive disease. It starts out with obesity and insulin resistance, and it takes quite a bit for it to progress all the way till somebody actually gets diabetic. But this is where physicians spend most of their time when it's already too late. The ideal case would be to have the prevention start right there in the beginning. But the problem is that we're limited by diagnostics. Currently, BMI and family history are used, which are not really the most sophisticated measures. So static measurements really don't cut it when type 2 diabetic is developed over a long period of time. So incorporating a temporal element is really critical. Hence, we see there's a clear and eminent need to develop new diagnostics and biomarkers for type 2 diabetes. But why is it so hard, apart from the fact that it is a complex metabolic disease? First off, human data is typically very noisy. To give you a small example, in 2005, in a paper, the authors could not find even a single differentially expressed gene under statistical significance between groups of people who were type 2 diabetic and those that were not. And this is not uncommon for human data due to sample variability. Recently, through GWAS studies, many novel SNP associations have been revealed with type 2 diabetes, which is good. Primarily among them is this particular TCF7L2. This transcription factor is also known to play a role in Wnt signaling. And I would urge you to remember this little connection between Wnt signaling and type 2 diabetes for the purposes of a climactic ending to this talk. Overall, these variants really don't explain much of the disease. So we need something which is more generalizable. So why do I use systems biology for this? So our lab looks at global snapshots of different models of insulin resistance across mouse and human and cells. This generates a multitude of data and challenges to integrate all of this data from different platforms to answer this one overriding question, which is what causes insulin resistance and its eventual progression to type 2 diabetes. And as a systems biologist, my aim is to look at the metabolic disease as a whole and integrate data from different models and platforms to answer this very question. In my talk today, I will present part of my integration work where I investigate two global scale transcriptomics data in mouse and human to find a set of genes that might be responsible for incidence of insulin resistance and obesity and eventually investigate their clinical and biological significance. The integration work that I did was done in 3 different stages. And this is specifically designed so we can find information that is not apparent from the prior levels. So models of insulin resistance in mouse and in human are quite comparable. Hence, using the readily available mouse model as a guide at each step, it not only helps me to enrich the human data, but also helps us gain statistical power for analysis. This brings me to the mouse data. So we were looking at muscle tissue of mice fed a high-fat diet over a time course of 6 weeks. The genetic profile of these were compared to those that were fed a chow diet. From the whole body glucose tolerance test down here below, you can see right at 5 days, the body is rendered insulin resistant. So first, I performed a very careful temporal analysis to sort out the initial drivers of insulin resistance in mouse, and then I used those results of the temporal analysis to integrate with the human data. What did we have for the human data? Well, we had about 80 subjects divided into 4 groups, that is obese insulin resistant, obese insulin sensitive, obese type 2 diabetic, and lean. And there was even a further subset of 7 patients from each that were sent over for genetic profiling. So in the end, we had about 28 samples of human muscle biopsies that were sent for gene expression analysis. You might also note that this particular group is very interesting, which is obese but insulin sensitive. So it'd be really cool to find out what's going on there. So I'm going to start with single gene level analysis and how I integrated the mouse and human data. So I used the genes that were found to be differentially expressed in the mouse analysis, and the most significant ones there got a higher weight in the human analysis. This plus a standalone analysis in human gave me about 40 genes that were differentially expressed at the single gene level. So each panel here actually answers very distinct questions. So through the red and the blue, I show genes that were up or down regulated respectively. For example, when you're comparing type 2 diabetics or obese insulin resistant people to obese insulin sensitive group. So we can actually isolate genes that leads the progression to type 2 diabetes from insulin resistance, because obesity in these are controlled for. At the gene set level, what could we do? We know that genes work as a group in systems to make a change. So why don't I look at that? I created my own gene sets of tightly co-expressed genes across the time course in the mouse muscle data, which I show here. So this is the time course, and you see the clusters. And I found 10 clusters, and I looked at these clusters as a group in the human data to see how they change. If in human a group was significantly altered, that is up or down regulated, I added them to the group of genes of my previous 40. So in all, now from 40 from the previous level and 80 from the gene set level, we have 120 genes that were differentially expressed. To do a pathway analysis was a good validation, because as you see, the top hit was insulin signaling pathway. So this was great, and we also found a few other interesting ones which are being investigated further. What did we do to validate further? Well, I compared my set of 120 genes that I show in dark gray here with a random set of genes that I picked in light gray, and I looked at their overlap with the hits from other screens that were performed in the lab. And as you can see, there's a clear and superior enrichment of the 120 genes with the hits from other screens when compared to the enrichment of the random genes. And the second half, I looked at some external microarray data that I downloaded and analyzed for human that were relevant to type 2 diabetes, and I wanted to see if this gene set of 120 actually alters in those data sets or not, and it does. A little interesting and slight different angle to this thing as well, could any of these genes really improve prediction of the disease status of these 28 people that I was looking at in the clinics? To find that, I looked at the 120 genes in the 28 subject and fit a feature selection algorithm to find the genes that were the best predictors of the disease status of these 28 subjects. And once I found that, I picked the top ones and I added them to the larger clinical data set of people with like 90 or more metabolic measurements. And now I wanted to see if I could still pick up some of these genes to be strong predictors of the patient outcome. And what we landed up finding is that, yes, actually a few of the genes do get picked up along with other metabolic features which are sort of known, like glucose intolerance, etc. This is a work in progress, and a lot of optimization needs to be done, but at the same time, I just wanted to show you what's possible. Moving to the third and final level, which is the network level. Here, I wanted to find two things. I wanted to see if I could further prioritize my targets from the 120 to a fewer set that we could actually drill down in the lab. And the second is, I was curious to know about the topology of interaction of my set of 120 with those of the insulin signaling pathway. What I found is there were 9 that you see in green in my 120 that were overlapping with the proteins in the insulin signaling pathway. But the remaining 111, what's going on with them? I wanted to know if the remaining 111 that were not part of the insulin signaling pathway are any of those, that single gene linker that is very extensively connected to the members of the insulin signaling pathway, enabling crosstalk between the pathway it belongs to and the insulin signaling pathway. And to find this, I thought this kind of visualization really helps, where you can go like on a gene by gene basis and look at how many other genes it's interacting with. And using this sort of a tool, I'm going to show you is just a few examples, is this highlights one of the most connected proteins from my set of 120 genes, which I show here, highlighting JAK1, and its interaction with all the proteins of the insulin signaling pathway. So the red line shows the connections, obviously. So what we're looking at is the genes that have really thick lines coming out. And the other example I have is for beta-catenin. And you can see how heavily connected it is. So we actually picked 4 targets from the previous protein-protein interaction map that I showed you, and we wanted to validate them. To do that, we looked at insulin-stimulated glucose uptake assay in muscle cells. So in the control case, as I show here, under normal insulin stimulation, we see an increase in glucose uptake in the cell. But when a protein, such as, let's say, beta-catenin or JAK1, is inhibited or blocked under insulin stimulation, and if we observe a defect in this glucose uptake, then we know that it is involved in insulin signaling. And I'm going to show you just 2 of the data because of lack of time, but we actually landed up validating all 4 of them. And what you do see here is this is for beta-catenin and this is for JAK, is that upon inhibiting them, we do see a defect in the glucose uptake. And if you remember my reference to Wnt signaling before, beta-catenin here is actually an integral member of the Wnt signaling pathway. And isolating this protein as one of the most linked one to the proteins of the insulin signaling pathway could potentially be very interesting in teasing out some of the inter-pathway and inter-disease relationships. So the final open-ended question sort of is, could these be the important segue proteins linking insulin signaling pathway to other diseases? And we have to do a lot more investigation to answer that. And that brings me to the conclusion and summary of my talk. If you look at it, what I'm trying to show here is through the integrated information that we get from a model system such as mouse in 3 different levels, that is single gene set and network level, we are able to enrich the human data and improve our analysis power. This shows that the choice of methods we choose to implement in investigating the data really does matter. Also, that different levels, that is gene set and single gene network, gives very distinct information. For example, without the network analysis, I don't think we would have come up with the targets that we landed up validating. And this enriched set of genes that we found is going to be optimized further to see if we can add more value to clinic-based diagnosis for a patient's disease status. And with that, I would like to thank a few people. Primarily my boss, Professor David James, who's been an awesome boss, and Dr. Jean Yang for all her statistical brilliance and in guiding me through the projects. Paul, who did most of, pretty much all of the validation work for the targets in our lab, and Catherine, Dorit, and Jerry for painstakingly collecting all the clinical data for the subjects. And thank you.
All right, any questions?
I would not be able to answer that question whether they were checked back later. I know we're doing follow-up studies on them, but this was a 5-year study, and then it's an ongoing process, and we haven't got the data from the new data, but it is being continued, so I would be probably able to answer that in some time.
Just a question with regards to your network analysis. One of the theories out there at the moment is that the least variant genes are more likely to be hubs. Does it surprise you that your differential expression marked genes were hubs? And how many of the other genes weren't actually hubs and were passenger genes?
So the interesting bit here is that none of the ones that turned out to be hubs were identified at the single gene level. That's what I would call as a gene, which was differentially expressed significantly. So the ones that I picked up were as a group. So it doesn't mean that, like JAK1, for example, was changing a lot on its own, but as a group interacting with a bunch of stuff, that's when it was picked up in the gene set level. So it kind of, I think, follows what we know from literature, that it's not really superbly changed on its own, but as a group. So.
Okay, thank you.